the title of his presentation today is Freedom of Research, CRISPR-Cas Editing in the European Union context. Yesterday, we will discuss, analyze broadly the legal and ethical framework in the field of scientific research, scientific experimentation at the international, constitutional, and European level. Today, we will experiment it in a very key context where all the issue, all values we discussed yesterday must find a reasonable balance. So, Iñigo, the floor is yours. And bienvenido.
Okay, tante grazie, Simone. Thank you so much. So first of all, let me thank Simone for the introduction, for inviting me to be with you today. It's just a great pleasure with a topic that I really love and I think that you could find interesting. Let's try to make it interesting at least. So if it makes sense to you, I will just share my presentation and we will be talking today, as Simone is telling you, about gene editing. This is a very interesting technology. The point is more or less that one of the things that probably Simone told you yesterday is that in general, freedom of research is a fundamental right, and it's got two different meanings. On the one hand, it means that researchers are allowed to perform any kind of research that they consider particularly interesting, or just they want to do that. And in principle, if you don't have a nice reason to oppose against this kind of research, then that should never be forbidden. And the second is that in general, all of us, I mean, people in general, the citizens, our societies, have a right to benefit from scientific research. So that all of us more or less have a right to have a nice research and benefit from everything that comes from this kind of research. So that's more or less what we have. And this idea of freedom of research is particularly challenging in the case of the CRISPR-Cas editing technology. So I think that the best thing that we could do as a start, and I think Simone, this will be the most convenient, is that we could start by showing a little bit about the technicalities of gene editing, what it is, how it is done, and why it is so easy to do and so important. So maybe, Simone, you could just launch the video and everybody will see this very short explanation about CRISPR-Cas technology for gene editing that I think it's really necessary to understand how it works. It was a very useful presentation of what the CRISPR-Cas9 technology is. Indeed, I will use this video usually because I think it's the most descriptive and easy to understand of those that I know, and it's got subtitles. That is always an advantage for whatever might happen, and especially for those who are not natives in English. So I really hope that you understood it. Just to make an update on this, as they were saying in the video, this technology can be very useful in order to face lots of different diseases, but also impairments typical from the human being. And indeed, we are already using that. And it's being used in two different ways at this moment. One is that we are, for example, extracting cells from a human's body, and we are just changing the DNA of these cells, and we just reinsert them in the human body. We transfer it again, just to try to solve some particularly resistant diseases, such as, for example, some types of leukemia. So this could be done. And lately, we are also trying directly to cure some disease that are present in the human body, especially some diseases that have to do with our eyes, with our vision. So last experiments, last clinical trials are related to diseases in the eye, and we are already trying to solve these kind of issues through a direct transfer of DNA in the eyes. If you wonder why we are doing this in the eyes, it's just because it's the one of the organs in the body that is not so well connected with the rest of us, meaning that if by any chance there are some kind of a bad consequences, you will just lose one eye, for example, but you will not lose your life. Instead, if you this is just because the problem will remain just in your eye, but will not extend to the rest of your body. Instead, if we try to make any changes in the DNA of one of your kidneys, for example, this could quite easily spread to the rest of your body, and this will make a huge issue. So that's more or less what we have now in terms of how we are using this technology in the human being. As the video was telling you, there are also some other applications that are very interesting and important in terms of vegetables and in terms of animals. Just to make you an example, we are trying to create mutated mosquitoes in order to face issues related to malaria, for example, that, as you know, is a very important disease, and lots of people suffer from malaria all over the world. So we are just trying to create unfertile male mosquitoes in order to end up with the populations of mosquitoes that really spread malaria. So this is one of the utilities that's been thought. And of course, we are creating lots of different enhanced animals for some different applications in agriculture, in farming, and some vegetables in agriculture. But I will especially focus today in the issues related to human beings. Okay, so I will just share again my screen, and I will try now to tell you what is the situation that we have and what are the main challenges that we have. So I will start with what we could call the controversial issues from an ethical point of view. I mean, the reasons why, in principle, we could limit the freedom of research. Okay, that's more or less what we have. I would just tell you in advance that in general, those will be issues related specifically to any changes in our genome that are affecting the germline gene editing. For those who are not familiar with that, germline is the type of genes that are really present in us, but that we also transmit to our descendants. So are those ones that we inherit. Okay, that's more or less what is the germline. So whenever we are changing the germline, it means that we are not only changing ourselves, but also our descendants. So that's more or less what this means. So somatic instead means that you are creating a change that will affect you, but not your descendants. For example, all those changes in the vision in the eyes are in the somatic line because they will never have nothing to do with your descendants, but only with you. Your children will never be affected by this, okay? So that's more or less the distinction. And the most problematic one is clearly the types of changing that have to do with the germline gene editing. So there are a number of reasons why it seems plausible and even recommendable to try to limit the research and moreover the application of that research in the human being. And those are the ones that you could see there. For example, risk, futility, vulnerating the sacrality of the human genome, vulnerating individual identity, the slippery slope to eugenics and the idea of enhancement, and the idea of gender balance, for example. So I will just try to explain these factors, and then I will just, in the last part of this presentation, I will give you some panoramic vision on how we are dealing with them at this moment at the EU level from a legal perspective. So I will just analyze what we have from a legal point of view in order to face this new technology. So I will start with this, and I will start with the idea of risk, of what happens with risk. Just to give you an initial tip, the idea in general is that lots of people consider that germline modification through CRISPR-Cas involves a very serious risk, and keeping in mind the precautionary principle that more or less states that when you have a serious risk, that is more or less easy to consider, that really seems plausible, and you don't know how to deal with that, it might be a good idea to avoid that kind of technology. And this is more or less what the European Group of Ethics has already stated, and says, well, the EGE is of the view that there should be a moratorium on gene editing of human embryos or gametes, which would result in the modification of the human genome. Well, so this is more or less what lots of people say at this moment, and I think that it's quite reasonable, in the sense that this is a quite recent technology. The video does not tell you, but we really started with that like not even 10 years ago. It was in 2012, if I remember it well, when we first started using this technology, when the first researchers that were afterwards that obtained the Nobel Prize, it's Charpentier and Doudna in California, started with this technology. So only 10 years is not a lot of time. We don't know so many things yet. And we know that sometimes the main problem is that we have two different kind of issues. One is what we call the off-targets, meaning that we try to replace a concrete part of a gene, but it happens that accidentally we change some other parts of our DNA, and this could lead to catastrophic consequences. And the second thing is that we really don't know so well how our DNA works. To be sincere, we only have a nice map of our genome from 25 years now. So there are a lot of things we don't know. So it's very difficult to mend things when you don't know how they work originally. So we have a lot of issues, and it's true that we have a risk in there. And so the point is that in general, this is a good argument to say that, hey, pay attention to this. But to be sincere, this works quite well when we are thinking about therapy, especially. I mean, if we are considering using these techniques in order to try to have better descendants, in order to try to start changing the DNA in the human beings, in adult beings, and so on, yeah, it makes a lot of sense. But when we are thinking about freedom of research, I don't think these arguments work so well, because precisely what scientific research is trying to do, always tries to do, is try to solve these kind of issues. If you consider that there's a risk because we don't know enough things right now, I think that the worst thing that you could do is to forbid research because of that. Because it will be like, just like when you ban your child not to start cycling because he doesn't know how to cycle. Yes, of course, but if you ban it, he will never know how to ride a bicycle. So it's totally absurd at all. Okay, so I dare say that the question of risk will be even a kind of a argument supporting scientific research in so many ways. Okay, of course, with some limits, because it's not the same if you are just introducing changes in embryos or that will never be transferred now in a woman's uterus, or whether you are considering another type of research. So the question here will not be if we are limiting scientific research, but what kind of research is now the most promising one, and the one that really deserves approval. So that's more or less what we have. And we should never forget something that is common to all types of scientific research. A certainty never exists. Just to be sincere, and Simone is a great expert in this, we don't even know if in vitro fecundation has consequences in the very long term, because we have never been able to see what happens with people that has been created through in vitro fecundation in the very long term, because no one has really been alive for a very long term if he's been born by in vitro fecundation. I don't know when it happens the first time in Italy, but I guess it was at the beginning of the 80s when the first Italians were born through in vitro fecundation techniques, meaning that they have now they're like 4 years old now, meaning that we don't know what might happen with them when they will be 70 or 80. because nobody is in those years. But this doesn't mean, this has never meant that we could not make research in that. I mean, risk will always exist and uncertainty will always exist. So we always try to do our best. And what we try to do is to try to make research in order to precisely reduce the risk. So that's what I could say about this factor, about the risk, of course. And then we have something that is called usually futility. What does it mean? Futility is usually that some people are saying that this kind of research is futile because it's not really solving any issue. So maybe it could be acceptable if you just want to gain some knowledge, just to get some knowledge, but will never be needed for the human being to have a better life. Okay, so that would be very important, for example, for the allocation of funding for the research. Because if you are making some research in order to satisfy your curiosity, that's fine, but please don't ask for public funding. Public funding is only allocated to our research when we consider that this could be part of the public interest. Okay, so the point here is some people are saying, no, we don't need this, because if we want to avoid to have some kind of people, and we want to avoid some especially important genetic diseases, it's enough with what we usually call pre-implantation genetic diagnosis. The implantation genetic diagnosis is a technique that is now being used in vitro fecundation, just in order to check whether some embryos created through these techniques have some kind of very important diseases. If we take a look at that and see that those diseases are present in those embryos, we never transfer those embryos. So in principle, that technique is enough in order to try to avoid some very terrible diseases such as Pais disease or Huntington disease or something like that. In general, all of those are monogenic diseases, meaning that they only have to do with one concrete gene. So this is a nice critique in the sense that it's saying just something like, right to research does not really apply so well, especially in terms of what we usually consider the kind of the right of people to benefit from science. If some kind of research is futile, this does not apply. I don't think that this is the case, because first of all, pre-implantation genetic diagnosis is not really the same as a gene editing is, because what we really do with the PGD, with pre-implantation genetic diagnosis, is something quite similar to eugenics, to be sincere. It's not that we cure embryos, to say so, it's that just we discard those embryos that really show a kind of disease. Instead, with CRISPR-Cas, what we do is to change the genes that launch that kind of disease, that trigger the disease. So we are somehow curing those embryos, and we could transfer those embryos, not to throw them away. So it's quite different. And in the other way around, it's more interesting. We could just say that this kind of research and its applications could apply to a list of diseases and uses that is much more extensive than just diseases related to a concrete gene. And to make you an example, we could, for example, with this kind of techniques, change several genes at the same time. One of those could be the BRCA genes. BRCA genes, for those who are not familiar with them, are the kind of genes that trigger some kind of a cancer, such as, for example, breast cancer. This was the type of genes that are usually screened when you are looking for what's going to happen with you. For those who, maybe some of you know the case of Angelina Jolie. Angelina Jolie had a preventive mastectomy because her BRCA genes show that she had a very high probability to suffer from breast cancer. So the point is that in the future, we could use this kind of a technology to change the expression of these BRCA genes so as to really mend it from the very beginning. And this is something that we could hardly do with pre-implantation genetic diagnosis. So the accusation of futility doesn't seem to work so well. Then we have another different thing, that is the argument of the vulneration of the sacrality of the human genome. This is a very familiar argument for those who really work on these kind of topics, because that's been a lot of times when lots of people consider that what humans do, it's wrong because it goes against nature. Okay, so somehow there's kind of supposition in there that a nature is something good, that nature works very well, and we human beings are not really making a very nice decision, a very smart decision when we decide to alter the way in which a nature works. Well, this argument is quite important, because if we really accept this argument, we should end up with any kind of a research that has to do with the germline, because it's impossible to use this kind of research in practice in embryos without changing the way things work. So we would probably be changing the natural mechanism of reproduction. Is this terrible? Well, I'm not so sure about that. I mean, it really depends on a different of things, of ideas. One is that first of all, the human genome as such, something that it's not something that is fixed. I mean, our genome is constantly changing. It's nature that usually changes it. And for those who are familiar with the evolution theory, the changes that nature introduced in a DNA of our descendants is really chaotic. It's not something that it's planified or anything like that. It's just that nature introduces a random, like 70 changes in each generation in our genes, and we just see what happens with those changes. If those changes work well, the descendant survives quite a lot and is able to have his or her own descendant, so the change remains. If the change is not working well, then this one really dies before being able to have some descendants, and the other gene disappears. That's how evolution works. So what we are doing here is substituting a quite chaotic and blind mechanism for a let's say a human thought mechanism. So the point here is that we should only ban this kind of research if we will be considering that somehow nature is more intelligent than us, that nature knows much better, and we believe, for example, in the idea of Mother Nature. To be sincere, I'm not really a great supporter of this argument, especially because after living, a pandemic, I'm not really so confident in nature. I'm quite unconfident. I mean, nature is quite neutral. Sometimes it will protect us. Someone will have to protect us against some of the things that may happen by nature. So in that sense, if you don't really believe that there's a kind of, intelligence in nature, or a kind of God that is working through nature or anything like that, it's difficult to really consider that human beings will necessarily be worse than some kind of mechanism that might be there but it's somehow blind. So that's an argument that is quite weak if you don't really believe in some things. Then we have the idea of the vulneration of identity. That is very, very interesting. Why is that? Well, the argument here is that this kind of research that we are performing, if it ever is used in a human embryo, for example, will be changing some very important functions in this embryo. And this could be so important that we could be changing the identity of this embryo. Is this important? Well, I don't really know. I mean, there is something that we have already been doing for some time, and we have allowed that. And for example, in the United States, it was quite famous, the case of a couple of lesbians who were deaf and were willing to have a descendant who was also deaf, because they were considering that that was not really a disease, but a kind of a way of living, being deaf, just to be sincere, and they really managed to do that. So somehow they consider that it is part of their identity being deaf. Well, to be sincere, I'm not so sure why it's part of our identity. I don't know what really makes us be us and not anyone else. For those who don't know so well about philosophy, identity is one of the classic problems in philosophy. It's very old. An identity, to be very concise, identity is what makes you be yourself and not no one else. That's what creates your identity, okay? And it's true that in our societies, it seems that identity is being very, very important, or it's becoming very important for so many ways. But I'm not so sure on whether this makes sense or not. For example, I know this is very Anglo-Saxon and very American when they tell you, yes, be yourself, no matter what they say, okay? That's nice if you are a nice chap. If you are not that one, I don't think that preserving your identity is so important. I mean, I know some person that if I had to give them a nice advice, I will just tell them, no, please change your identity, really change it. I mean, you will not lose nothing. I mean, the world will be better with a different you, and you will feel better being someone else, but not you, okay? So in some cases, preserving your identity is not really the best idea. And this is particularly true if, for example, you will suffer some kind of disease. For those who know that, Alzheimer's disease is one of the type of disease that usually is considered to be identity changing. Indeed, those who are suffering from Alzheimer's usually says, or the people who are with them say that they are not themselves anymore. They are still the same biologically they are the same person, but they are not at all the same moral agent, to say so, or the same person, because their conscience is lost. So if we were able to revert that process and send them back to the former identity, that will be changing their new identity, but I don't think that it will make any issue. So I think that that it's not a good idea to forbid this kind of research only because it has to do with this sense of identity. There's another good argument on that usually state something like, well, you are convincing, and it seems that indeed we could more or less accept that this research is morally acceptable, it's well grounded, when has to do with therapy or when has to do with something like trying to prevent some kind of diseases, or when it has to do with anything like that. I mean, but what is not acceptable and should not be acceptable is that we use this kind of technology for some kind of eugenic purposes or for enhancement. What does this mean? Well, those people are saying more or less that you could use it to have a healthy descendant, but it should be immoral to use this technology to have, for example, a clever descendant or a stronger child or a faster one or a prettier one or whatever. This is what we usually call enhancement. And there are a lot of people who consider that those both things are different. And I think that they got it right, because they are not really the same. And just to be sincere, this has to do with the differences in what we usually call absolute good and position goods or relative goods. To make it quite easy, a health is usually an absolute good, and it's not related to someone else's health in general. Meaning that if you are healthy, good for you, but it has nothing to do on how I'm feeling. If I'm feeling bad, that has nothing to do on whether you are healthy or not. Instead, in something like, for example, strength, you are not usually strong or weak by yourself. You are always strong or weak by comparison. If I am with Simone, I will feel more or less, well, not so bad, not so strong, not so weak. If I am with Sylvester Stallone and Arnold Schwarzenegger in a room, even if they are in their 80s right now, I will feel quite weak, to be sincere. And most probably lots of you will consider that I'm not so strong. So if by any chance we start to make some of our descendants stronger, then those who are not really have not received this kind of enhancement will seem to be weak in so many ways. This will be the same in the case of intelligence and in the case, for example, of beauty. If we all start to be like Brad Pitt or something like that, well, most of us will really become some kind of ugly people. So that's why I'm telling you that they got a good argument in that, and it seems to make a sense that we just limit the use of these technologies to some concrete functions. But the main problem is that those who are using this argument usually go one step further, and they say, yes, in theory this works well, but in practice, when we start using that, it's very difficult to trace boundaries. Because if people know that from a scientific point of view, they can have a stronger, more intelligent, a more beauty descendants, they will probably use this technology for that purpose. And I'm quite sure that they also get a point in this, but it might happen that we are able to introduce these boundaries. So it's not so easy to know whether we should be banning all kinds of research of this type only for this reason, even though it is quite an important reason. And I will also start or finish with some other important reasons that we should consider before going on with this kind of research. And I will again remind you that one of the things that is necessary in order to protect the freedom of research is whether this will really benefit the society or not. And when we think about gene editing, we should always consider about the type of society that we will be building if we go on with these techniques. This is very important. Now let's think about the future. Let's think about the society in which it will be really possible to change the genes of your descendants and see what will happen in that society. Just to introduce you a fundamental factor to consider what will happen, we should consider that this technology, even if it is very cheap, it costs not a little, just like $40, $50 to change someone's genes. This is very cheap, but it can only be effective if the embryos are in a Petri dish. So the point is that we should use in vitro fecundation in order to be successful if we want to change the genes of our descendants. In this case, changing the genes would only cost $50, but using in vitro fecundation technology will cost at least $30,000 or something like that. I don't know how much it costs in Italy, but more or less this is the standard cost, $20,000, $30,000. Even if it were cheaper, this means that not everyone could really use this technology, meaning that only unless the public system would afford something like this, only those ones who will be more or less rich enough to pay for the money will have really healthy descendants. The other ones will not. This doesn't work so well in terms of equity, of social equity, okay? This, as I was telling you, this not necessarily will have to happen because we could, of course, offer some kind of public resources to do that, but it's not so easy. And I'm not so sure on whether we could with that. And even in this case, there will be some people that will not be really so happy with using those techniques for having natural descendants. And what's more, I guess that you all know that it is not always that we have children because we were just trying to have children, but sometimes it just happens. You do something, and at that moment, life starts, and it was not planned, but you follow up with that. And those who will be born through these techniques could be somehow seen by the rest of the people as poor people who have not been, ensured a quite healthy life. On the other way around, this has to do with what we usually call the procreative beneficency principle. That means that in general, we parents always try to do whatever possible to have the healthiest possible descendants, meaning that if we, for example, can protect our children by giving up smoking through pregnancy and so on, we have to do that. And if we will have a better children by, for example, changing some genes, we are supposed to do that. But of course, this will introduce a lot of pressure into couples. And in the sense that we will feel like, well, I really should have my children, by in vitro fecundation in order to have the healthiest child that I could ever have, because I owe these to my future children and so on. And there will be a lot of social pressure in that. And this pressure will especially affect women. So there will be some kind of gender issue in that. And this is due to a simple reason. For the males, it is not really so constraining to go into a kind of in vitro fecundation process, because the only thing that we have to do is just to, to donate some sperm. And this is not really so terrible at all. We can give hundreds of millions of spermatozoes, and this is not really a painful procedure that we need to carry on to do that. But for women, this is very difficult. They have to carry on a kind of of stimulation, and this is a quite hard procedure, risky, sometimes painful, and psychologically is also quite hard. So it's not so easy to do that. But they will be much more pressure to go for that than males, because we somehow always have also what I usually call the idea of the good mother. And the good mother is especially expected to do the best for the children, so the pressure will be very, very strong for the women. So all these factors had to do with a key issue that we always have to keep in mind when we are considering whether some kind of research should be allowed or not, or what are the boundaries of the right or the freedom to research. Because if we really don't think that this will really benefit the society in the future, if we consider that this could be indeed something terrible for society, then it will be much easier to introduce boundaries or controls in the exercise of the right freedom to research. So that's more or less what we have from an ethical point of view. And now I will give you some really quite short ideas about how we are regulating this right now at the EU level. So let's go for this, that really will be kind of bullet points. So first, we have the Charter of Fundamental Rights of the European Union, that, as you know, it's legally binding right now. And we got Article 3 that recites, in the fields of medicine and biology, the following must be respected in particular: the provision of eugenic practices, in particular those aiming at the selection of persons. So we have a general provision of eugenics at the EU level that is partially applicable to gene editing. So in theory, we should not use this gene editing to create somehow like enhance descendants, but it's nothing to do with health. Then we have the next one, that is what got one step further. It's what we usually know, the EU Directive on Biotechnological Inventions. This has what we usually call the moral clause in its Article 6, that provides that inventions shall be considered unpatentable where their commercial exploitation will be contrary to all the public or morality. And the paragraph 2 of this article continues on the basis of paragraph 1. The following in particular shall be considered unpatentable: processes for modifying the germline genetic identity of human beings shall be considered unpatentable. So, I mean, what does this really mean? Well, nobody really knows, because here they are just mentioning processes modifying the germline genetic identity. Well, this is very difficult to understand, because nobody knows what genetic identity means, as has never been well described nowhere. So the problem that we have is that in practice, we don't know what is really forbidden or what is unpatentable, keeping in mind this clause. As you will see, this is a real issue, because the other normative tool that we have, that is the EU regulation on clinical trials and medical products for human use, well, has an article number 90, that more or less uses the same concept. And it states that clinical trials with gene therapy medicinal products that cause changes in the gene identity of the person's germline are prohibited. But the problem is more or less the same. What happens with that? But we have an additional issue here. That is that in principle, when we are considering this type of trials, it seems that they are just referring to what we usually call persons as such. But Simone can correct me if I'm wrong. In general, we don't consider human embryos to be persons, nowhere in this world, maybe in some countries in Latin America, but not at the EU level. So whenever we perform these trials without transferring those embryos in a woman's uterus, this could be done at the EU level. So it seems that we could, but we usually don't call them clinical trials as such, because clinical trials means something that is like being used for kind of therapeutical use or something like this. So I think that we will be talking about basic research or some kind of research that is not a clinical trial, when we are just trying to know what happens if we are changing the germline in some embryos in a petri dish. And so we have finally the most important tool, that is the Oviedo Convention. The Oviedo Convention is an international treaty. It's not only at the EU level, but it really applies in lots of EU countries. I guess, well, Italy was a signatory of the Oviedo Convention, for example. I guess that you never ratified it, or you ratified it, Simona, you never know. It's very Italian, you signed it, but never ratified it. So it was very good in so many ways, because you made the photograph, but you never really, subscribed it in a practical way. But lots of EU countries really did it. Those that are exceptions are mainly Germany and the United Kingdom. Well, the United Kingdom is no longer an EU country. It's become a little bit Italian in some ways, but it's not an EU country right now. So the point is that this one is really enforced in a lot of countries, especially at the EU level. And we have a very nice article there, Article 13 in Oviedo Convention, that states that an intervention seeking to modify the human genome may only be undertaken for preventive, diagnostic, or therapeutic purposes, and only if its aim is not to introduce any modification in the genome of any descendants. As you can see, this is a very complex clause, impossible to understand, of course, totally impossible. This was created just to generate some chaos, in my opinion, because as you can see, first of all, it's something that is very well in the law. That's a kind of a permission, not a ban, because it is just saying that some kind of a research can be done, because it states properly that some kind of intervention can be undertaken. So it's a permissive clause, something that is very well. Usually our laws do not allow. It is usually considered that when something is not banned, it is allowed, that's all. So this is an article that is really allowing things. But in the way it's been redacted, it's of course an implicit prohibition of a lot of things. So if you read it in the other way around, it's something like, well, we will allow any kind of a modification of the human genome if it is undertaken for preventive, diagnostic, or therapeutic purposes, and if it does not introduce any modification in the genome of any descendants. So if these conditions met, well, we can go on doing this. So it's quite permissive. Main problem that we have here is that first, it's very restrictive, or it seems to be. And second is that it's very mysterious. It's very difficult to understand what the terms really mean. Because in this article, first of all, it seems quite clear that and most people think that if the germline is affected, everything is banned and you cannot do nothing with that. So if this is true, and if it does not change, it seems ridiculous to go on with the research, because it will never be used in our descendants, because it's banned by this article. But in my opinion, it is much more tricky than that, because as you can see, the only thing that is really forbidden by this article is a kind of intervention that is seeking to modify the human genome. But what is the human genome? This is quite a riddle in this clause, because the human genome could be defined in two different ways. First of all, it could be defined as the concrete a set of genes shown by a concrete human being. So I got a human being, a human genome, that is my genome, of course. But in terms of humanity, we also have what we call the human genome as such. and it's composed by all the different expressions of the genes that are shown by all the different members of the human family. So there are a lot of different expressions of the same gene in the human family. This is why some of us have very nice blue eyes, some very brown eyes, some got dark eyes, and so on. And all those expressions are human genome. Of course, in your genome, there will only be one type of expression, and that will be your genome. So if we understand human genome in the sense that it comprises all the expressions that can be found in the human family, if we change one of those expressions, that is, for example, a kind of expression that is linked to a disease, okay, an unhealthy expression, for a kind of healthy one, we will be changing an embryo's genome, but not the human genome, because there will be no novelties in the genome of the human being. Instead, imagine, for example, that we introduce a gene of an animal into a human embryo. For example, I can't remember the name in English, but you perfectly know those animals that are very nice, that really bright in the dark. I don't think it's lullaby, but it's one of these insects, you know, that really you just get some lights when it's night. And you think, for example, well, I don't want to spend my life looking for my children at night, so I prefer to introduce this kind of gene in my embryo, so as when they become children, I can really see them at night because they will have those genes and they will be brightened at night. Well, that will be banned by Article 13, because you will be introducing a modification in the human genome, because there are no humans in this world expressing that animal gene. So that will be banned. But if you are just changing, for example, the genome of your embryo in a way that it was showing a such expression, and now this is not showing that kind of disease, then you are changing that embryo's genome, but you are not changing the human genome as such. So what should you understand of all those things? Well, a very tricky thing is that whenever we are making bans that have to do with freedom of research, a very wise advice is try to use type of reductions that could find different meanings. I know that for those who are real lawyers and say, well, this is not a good idea because the law should be, you know, precise and perfectly tell you what is forbidden and what is not. So those ones will not really be probably accepting this. But in practice, when you get an international treaty that is very difficult to change, that will mark a lot of things and will affect fundamental rights, it's good to have some kind of open interpretations that could change depending on the state of affairs. One of the things that we have right now is that we don't really know what's going to happen with gene editing. So it's very difficult to make a bet in any senses. So this kind of clauses at some point might be useful in so many ways. And in any case, that's more or less what we have at the EU level. Of course, what we have is very vague, as you can see it, somehow obsolete in so many ways. That it's based on an ethical framework that is under discussion. And we need to reflect on that, that's pretty sure. And we need to have public opinion involved, because we all need to be aware of how much our societies might change because of our research that is being formed right now. So I think that the decision should be in our hands, I mean, in everyone's hands. But of course, we will always have a final issue that I would show you, is that world is composed by a lot of different countries, and the rule of law is not the same in all of them. So one of the main problems that we have is that if we ban some kind of research in some countries, there will be some other countries that will probably develop it. And this also makes a good point and an essential thing that we need to discuss. So I think that's all, Simone. I guess that it was enough. I think that I talked too much, maybe, and I will be very, very happy to hear your questions, your comments, or whatever. Okay.